Thanks ever so much. Over to you.
Great, thanks. I hope you can see the shared screen and pointer.
Yes, both.
Okay, great. So so I'm going to tell you mainly about methods, and it was actually a great segue from the previous conversation about identifying fine cell types, not just with single cell sequencing, but with in situ sequencing. So why should you care about fine cell types? The brain contains thousands, and that's a conservative estimate, thousands of cell types. And even a single brain area contains hundreds of types of just neurons. Why does that matter for neurodegeneration research? Well, different types of neuron do play different roles in computation. And it's possible that the cognitive effects of neurodegeneration are due to specific cell types. And I think the best example of this is Parkinson's disease, where the vulnerability is really in one very fine, one cell type, the dopaminergic neurons, but just the dysfunction in that cell type can cause massive brain-wide effects. For other disorders like Alzheimer's, we don't know yet, but it's very possible, as emphasized by our own Karen Duff and many others, that different cell types are selectively vulnerable to neurodegeneration, and that the cognitive effects could come specifically from specific cell types. Okay, so I'm going to talk about mainly methods and in situ transcriptomic methods for identifying fine cell types. And as we heard this morning, there's amazing development of the technologies in this field every year, but still I think we face this quadrilemma. We can't have all four of the following things. We can't have high throughput, that we can do large scale experiments in a day or two at a cost that actually allows us to do a large number of these experiments on a regular basis, while multiplexing large numbers of genes at high sensitivity, as Wei Ting emphasized in the morning. So what I'm going to tell you about today is some work with a specific method, which is the padlock probe method, which is in fact the one you heard from Wei Ting that's now commercialized by Katana. And this method gets 3 of these, but not the 4th. So it can process centimeter-sized samples within a day or two. It's a very low cost per experiment. If you have the microscope and the DNA probes, then the cost per experiment is basically 0. So you can take advantage of the high throughput. It can detect hundreds of chosen genes, actually thousands. If you want to, we've actually found that you don't need more than 100 or so to do fine cell typing. But it does not identify every single transcript of the genes. That said, the technology is moving on, and something I'll tell you about today, it's now around 50 times more sensitive than the original method. Okay, so what I'll tell you about, first, experimental methods for localizing many RNAs simultaneously, computational methods that we use this to map the fine cell types and a verification that it actually works. And finally, some exciting new data connecting this to in vivo functional imaging, so we can see the activity of these cell types in the living brain. So the first part, the experimental methods, was a collaboration with Cheyenne Kian and Thomas Holling and several others in the lab of Mats Nilsson in Sweden, and it's published in this paper that you should be able to get to if you point your phone at this barcode. Okay, so the way the method works is you take your tissue section. In the original method, it needed to be cryostat cut. We've developed now, you can use thicker vibratome cut, thick sections. But you take your tissue, and the first step is a chemical step that turns the RNAs into complementary DNA strands. This is done in vitro. The next step is to wash on these padlock probes. So the padlock probes are strings of DNA that are custom ordered to target genes of your choice. So you select 100 or 1,000 genes of your choice, and you design these padlock probes such that the 2 arms of the DNA, the two distal arms of the DNA, target a sequence of interest in your target gene, and then hybridize to it. The next thing that happens is that you wash over the tissue a ligation enzyme that ligates this into a circle and then perform rolling circle amplification that goes round and round this loop, making a blob with hundreds or thousands or loads of copies of this string. And in particular, it's copied a barcode sequence, which is also on the padlock probe. So as a consequence of this, you can then read out through multiple rounds of fluorescence imaging, you can read out the barcodes and identify what barcodes they are, and therefore what genes were there in the tissue. So here's an example of some raw data from this original method. There are 5 rounds of fluorescence imaging, each of which uses 4 colors. And if you focus on this spot here, you can see it's yellow, then blue, then green, then red, then red. And that corresponds to a particular 5 base pair barcode, GTCAA, which, because we designed the padlock probes, we know corresponds to a particular gene, in this case, RGS10. So that's how the method works. Here's an example of the sort of data you get. This is a section of a mouse brain. These are the genes that were chosen. They're color-coded. So similarly colored genes live in similar cell types. And you can see here the hippocampus. If we zoom into this yellow region, we see further, there's a pyramidal cell layer. You can see CA2, CA3. There's some interneurons. If we zoom in here to stratum lacunosum moleculare, we're starting to see now individual cells. And if we zoom in again, we see now 1 cell. And so the first thing you'll notice is that there's 10, 20 so reads per cell. It's nothing like the total number of RNAs that are found in the cell. This is the inefficiency of the method. On the other hand, if you want to do fine cell typing, this is actually sufficient because this particular combination of genes, NPY, relin, KIT, NDNF, ID2, identifies the cell not just as a neuron, not just as an inhibitory neuron, not just as a neurogliaform cell, particular class of an inhibitory neuron, not just as one of those with a developmental origin in the caudal ganglionic eminence, but also a particular cell fine subtype of that, which is the subtype that does not express CXCL14. So even though the inefficiency is low, it's enough to get us down to very fine subtypes that account for, say, 0.01% of the total cells in the region. The technology, since that paper, we've been developing it here at UCL. There's a lot more going on. So we have a fully automated system. So it runs automatically. You put your section in, computerized fluidics and microscopy. You come back a day later, you have your data. It can now run on thicker sections, 50 micron vibratome cut fixed sections, which makes it much easier to apply the data using confocal imaging. New chemistry gives us higher efficiency, and new analysis algorithms, which is probably the most crucial thing, mean that we have much higher efficiency. And there's a brand new rig in the UCL DRI. So if anybody is interested in collaborating, please get in touch. Be very interested. So here's an example of the new method at work. This now is the cerebral cortex, V1 again of a mouse. If we zoom in, we see the individual cells, and we zoom in again, you can see there's many more reads. And if we compare the new method on the right to the original method on the left, we see we've got about 50 times higher efficiency. And this is still improving. So it's certainly good enough to be getting some good data. Oh yes, there was a question earlier of whether they can see vascular cells. And so I thought I'd put this slide in. This is a focus here on non-neuronal cells, and the vascular genes belonging to vascular cells in particular are shown in red. And you can see here, it happens to have sliced a blood vessel in the plane of section, and you can see all along the blood vessel, you've got this red color showing the vascular cells, and it starts to go out of the plane of section here. But you can see, if you look carefully, you can see the network of vascular cells, the blood vessel network revealed by this method. So absolutely, yes, you can see vascular cells. Okay, so the question though was about fine cell typing. Can we use this method to identify fine cell types? Because those fine cell types probably have importantly different computational functions, particularly when it comes to neurons. Okay, so we wanted to test it in a system where we know what the answer is. And there's one clear winner for the system to work on, which is the interneurons, inhibitory neurons of hippocampal area CA1. And the reason for that is that there's been 40 years of intensive effort put into classifying these inhibitory neurons down to their very finest subtypes. This comes from a Hungarian school of neuroanatomists. Shown here, Tamás Freund, Yuri Bajaki, actually my postdoc advisor, and Peter Somogyi, who was a collaborator with us on this project, here collecting the first ever brain prize in 2011 for this historical work, classifying these cells into very fine subtypes. So this classical work identified at least 23 subtypes of these inhibitory neurons. And I should say, the inhibitory neurons themselves only account for a few percent, maybe 5% of the cells in the region. Sorry, less, just a couple of percent. And then we've got many, many fine subtypes of these subtypes. At least 23 types of these inhibitory neurons are classically defined. And single cell transcriptomic data, not the spatial method, we looked at the fine subtypes of cells and found that it agrees almost completely with these classical results, with a few exceptions. For example, 3 of the fine subtypes that people had looked at with antibodies turned out to be identical because they had never used the same combination of antibodies together. And when they used different combinations, they thought they found different cell types. Okay, so here's a result from single cell RNA sequencing. And you can see, again, all of these cells together account for just a couple of % of the cells in the region. But within this inhibitory cell types, you have very fine subtypes. And I want to draw your attention just to one of them, this SST-NOS1 class, which is a very fine small subtype of that. It's only maybe 1% of the inhibitory neurons, which are themselves a small fraction. But this is the type of cell that could end up being very important for studying a disease like Alzheimer's, because these cells, it appears, are the ones that are responsible for causing blood vessels to dilate during sleep. And as you know, there are theories of vascular clearance being important for Alzheimer's. And so these cell types are just one example of the sort of fine cell types you really need to quantify. Okay, because the system is known so well, it's not just that subtype, we're able to understand what all the different subtypes do within the circuit. Okay, so how do we identify them in situ? So we use probability theory, and we start with a model for how the gene expression pattern is going to depend on the underlying cell types. And I'll go quickly through the math, but I do want to cover a bit of it. So the probability that you'll see a gene, particular gene G, coming from a particular cell C of class KC at a location X is given by this formula. It depends on the mean expression of that gene in the appropriate cell type, and that we get from single cell RNA sequencing. It depends on the position you are. It falls off as a Gaussian function from the cell center, and it also depends on 2 parameters. This is first a technical parameter, the efficiency of detection of that particular gene, and then also a random fluctuation that depends on the cell. So how do we identify the cells to classes? We use Bayes' theorem. We've got the probability of the gene expression given the cell classes. We use Bayes' theorem to turn that around and get the probability of the cell classes given the gene expression map. And actually we get more than that. We get the probability for each cell to belong to a particular cell class, but also for each gene read, each detection to belong to a particular cell. Just very quickly, I'll spare you the proof, but the formula that comes out of the math is that you essentially count the number of genes of each species in each cell, and then what ends up happening is that the cells are classified using a negative binomial model. So based on the in situ, the single cell RNA sequencing data and this efficiency parameter you estimate, the probability that you classify the cell as belonging to a particular class it follows Bayes' theorem with this negative binomial distribution for the cell, for the gene counts. Okay, so let's look at it working. So here's an example, and this is with the earlier low efficiency method, but you can see it's still going to work even with this low efficiency method. So here's an example of a zoom in into the hippocampal pyramidal layer, where the cells are very densely packed, and the symbols are colored. So green genes belong to pyramidal cells, and red symbols belong to this particular class of CCK positive inhibitory cells. It's a fine subtype of interneurons. And you can see that we've got a group of these red spots over here, and the green ones are elsewhere. In the background in white, you see DAPI, and the gray dashed contours are the boundaries of DAPI segmentation. And you can see the DAPI segmentation just can't really identify the boundaries between the cells, because the red spots identifying the inhibitory neuron are over here, and it crosses what appears to be the DAPI boundary. But because we use this algorithm that assigns the genes to cells as well as the cells to classes, it's able to discard the DAPI where it's not helpful and put the genes in the right cell based on the prior knowledge of which genes belong in which cell. And this prior knowledge comes from a database of single cell RNA sequencing that identified defined cell types to begin with. All right, so now let's see if it works. So because it's a Bayesian algorithm, it gives us a probability distribution for every cell type. And here you see some examples of identified cells, and the pie charts show the posterior probability distribution for those cells to lie in particular classes. We can get a spatial map coming out of this, and because we know what cells live where within the hippocampus, due to this 40 years of prior work, we can compare the distributions we get with what's known from the literature, and it works correctly. So because we know what cell types should be where, we know that we've identified the right cells lying in the right regions, and that gives us confidence that the method works. A very important thing when you've got all of this data is to be able to visualize it. So we put a lot of effort into this online data browser, and I please encourage you to go to this website, insitu.cortexlab.net, to see it. It lets you zoom around, see the different cells, see how they're classified as different classes and their gene expression profiles. So the last thing I want to tell you about very quickly, we're very excited about the connection to in vivo functional imaging. So these different cell types do different things, play different computational roles in vivo, and we'd like to understand exactly what they all do, and that will allow in the future things like understanding how that changes in animal models of neurodegeneration. So many, but perhaps not all of you know about this method of 2-photon calcium imaging. This is based on this molecule here called GCAMP. It's a protein that becomes fluorescent when in the presence of calcium. And because when neurons fire, calcium enters the cell, this sensor lets you see when neurons are active. The 2-photon microscope is a fantastic invention that allows you to image deep into brain tissue. And the way it works is this fluorophore is excited by blue light, but if you shine red light of very high intensity femtosecond pulses, you can see that the focal spot here is tiny compared to this broad point spread function you get in one photon node, 2 photon, because it relies on 2 photons hitting at the same time, only fluoresces at the exact focal spot. And that lets us do functional imaging of, millimeter square size parts of activity in the mouse brain with this GCAMP transgenically expressed. So here you see an example of this data. This is zooming out to this millimeter square 11 plane image, and the cells are flashing away, and they're flashing because they're active. When one of these cells flashes, that's because calcium's come into it. This is a black and white movie. It doesn't tell you what cell types are where, but what we can do is then take the brain that we imaged, cut it into slices, and then apply this in situ transcriptomic method. And here you see an example of this is the in vivo imaging Z-stack that we got with the 2-photon microscope in vivo. And this is a slice from it with the transcriptomic method applied. And you can see here the superimposition of GAD RNA, this identifies inhibitory cells, with the in vivo image of GAD and cherry, because it was a mouse in which the inhibitory cells were fluorescent red. And that lets us apply the in situ method to identify for the first time the different cell types that we've recorded. And we're really excited about this. I just want to finish by showing you one slide of the raw data that comes out of this. So this is just the inhibitory neurons, and they're in visual cortex. This raster plot on the x-axis is time, and on the y-axis is the neuron identity. And when it's black, that's because this particular neuron corresponding to that row was active at that moment in time. And the cells are sorted by their molecular cell type that you see over here. These top ones, neurogliaform interneurons, different types of inhibitory neurons, parvalbumin inhibitory neurons, the main class, etc. And if you look at this, you can see there's this incredible fine structure, which includes, amongst many other things, an alternation between 2 main subtypes of interneurons, the parvalbumin inhibitory neurons, and this one, the SNCG and LAMP5 neurogliaform interneurons. So we're really excited to see, using this technique, what sort of activity in vivo these fine subtypes have, and then in the future, how that can be changed in dementia models. Okay, so to summarize, you can localize very fine cell subtypes by localizing just a few hundred genes, and the method works on fine subtypes as confirmed by this acid test in CA1 interneurons. The efficiency keeps on increasing, and it can be combined with in vivo functional imaging. Okay, so thank you very much, and of course to the people who worked with us and our funders.
Fantastic, Ken. That's amazing. Beautiful images, but also I just admire the way you've really dug into some of the biology and helped to unpick. So I hope you've gone back to Peter Somogyi and colleagues and said, actually, 3 of your cell types were identical, so you need to hand your brain medals back.
Yeah, we had that discussion, and he took it very well. I mean, he was surprised, but then he went ahead and did some experiments which confirmed it, and was happily an author on their paper.
Oh, fantastic. Even better story. That's brilliant. So you've got this kit and it's set up. I mean, it sounds like it's a quite technical, elaborate setup that's in situ. So, is this something that you see yourself being able to condense and then roll out to other institutes, or do you see it basically as a centralized service where, because you're getting it better and it needs that sort of technical support, people will send samples to you?
Yeah, so in terms of collaborations with DRI labs, as I say, we've just set up the microscope. The person who is running it is Izzy here, and we've got projects we want to do with it, but we're also very happy to collaborate. I think the best way forward, because it is a finicky method still. So if somebody's interested in collaborating, I think the way to go forward is find someone in the lab who wants to do the experiments, and then we'll teach you how to do it. And I think that's the best way. A centralized service is a possibility, but we're not set up for it at the moment at UCL.
Right, brilliant. So Atticus Hainsworth says, lovely data. Karen Duff asked the question, can ISS be combined with tissue expansion? I don't even actually understand the question.
Yeah, okay, so tissue expansion is well, as you might guess, a way of expanding the tissue. It absolutely can be, and it has been. For me, though, tissue expansion is the opposite of what I want to do, because I want to image a large amount of tissue in a small amount of time. So when you expand it, you make it bigger, you need more tiles for the microscope. If you're interested in zooming right into a particular cell and finding the subcellular distribution, then that would be a good thing to do. But for this kind of larger scale project, I'm not sure that you need to do it.
Right. Blanca asked the question, hi Kenneth, could you explain in more detail how you managed to align the cells from the calcium imaging to the in situ data? I imagine it's not very easy to find the same cell again.
Actually really hard. And the reason it's so hard is because at least when we did these experiments, we had to take 15 micron thick, 10 or 15 micron thick cryotome sections. And just a little bit of distortion can make the match very difficult. But so it was really about algorithms rather than experimental methods for matching the two to each other, allowing for some distortion in the tissue. And it took a long time, but I think we've got it working. It's now going to get easier because the new methods will allow us to work on vibratome cut 50 thick sections, and I think it should be much easier in the future.
Can I ask, I mean, so you've got this, I'm very concerned about the concept of, which is, almost a human, it is a human, a statistical concept of a cell type specific gene. And, this is a prior that we rely on in so many of our methods, so many of our interpretations from genetic risk to kind of this data. Would your methods be able to detect, when, you are relying on a set of genes to infer a cell type in a particular position, would your methods be sensitive to detecting when the allegiance of one of those genes has changed in a disease state, for example? I mean, classic GFAP, and seems to now be a mark of activated astrocytes. So, how are you? Is that a fragility? Would you detect when your prior doesn't make sense?
Yeah, so that's a great point, and I think that's the reason to do a large number of genes in parallel, because if one or two, I mean, so in this class of inhibitory neurons, the great example is parvalbumin, which is the classical marker for a particular cell type, except it turns out that it's activity dependent. And so that gene on its own could lead you astray. But in combination with 20 others that are specific to this cell type, and I should say, no gene is found only in one fine cell type. It's always a combinatorial pattern. But in collaboration with the 20 other genes, I think you'd be on fairly safe ground that you're still getting basically the same cell type. But the question of if take that activated astrocyte versus a non-activated astrocyte, are they the same type or not? That's a matter of definition. Maybe they're a different cell type. Maybe it changed cell type.
It's a great point. That's a very good point. So there's a question from Jack Barrington, but I'll let you take that one in the Q&A because we need to push on to catch Phil's talk. So thank you ever so much, Kenneth.
Thank you.